So any questions you guys have about the material, we can go over certain questions or certain units, or any test taking or study tips for this specific exam.
So Peyton, in bacteria, we don't have gram-positive spirals, right?
No, I don't believe so. I'm not remembering any gram-positive spirals. I really think that the spirochetes are more gram-negative. Yeah, here, I'm double checking because I don't want to give you guys wrong information. But I don't think that there are any, at least not any that you guys need to know, or that we went over in this class. Bacteria, yeah. No gram-positive. It's just gram-positive cocci and gram-positive rods. So yeah, no spirochetes. And so that's helpful. So if you see something that is a spirochete, you automatically know it's gram-negative, is a good thing to remember.
And also,
Oh, go ahead.
okay, so like we have specific degrees in which like it heats up things, you know, like such as in autoclave or pasteurization. Do we have to remember these degrees?
The specific degrees, that's a good question. I feel like the more important thing to know is how it works. So you know that autoclaving, it's at a extremely high temperature. You know that it is a physical form of microbial, like control of microbial growth. So I feel like it's more important to know how they work and what kind of microbial growth they are, rather than the like super specifics about them. If you have time to memorize them, yes, it's always better to have more information than not. But I think the more important thing to focus on is specifically how they differ from each other, what each of them can destroy, things. Pasteurization. Yeah, no, autoclaving and pasteurization. No, okay, yeah, I'd say know the degrees for pasteurization. Yeah, I'd say know the specific temperatures for pasteurization. Maybe not for autoclaving, but yes, for pasteurization, know the specific degrees.
Okay, so I have two questions.
Yeah.
So you kind of answered it. I had messaged you about like how like it's microplasms.
Yeah.
They don't have like the categorization. So I was wondering, should I just leave it like as B, like its own little category since it's like special?
Yeah, they are strange. Mycoplasmas are very weird. They don't have a cell wall. They have sterols in their plasma membranes, but they still stain. And so even though they are different, they're discussed with gram-positive bacteria because they stain purple, because they don't have like I'm blanking again. But yeah, no, they're discussed with gram-positive bacteria because they stain purple. So even though they don't have a cell wall, it would probably count as a gram-positive bacteria because it does stain gram-positive. They're the weird ones though. Yeah.
Okay, so that one will be like its own category. Just
Yeah.
knowing that it's gram-positive and doesn't have a cell wall, and they pretty much just stain purple.
Yeah. Yeah. Mycoplasmas are quirky and special and unique and not like other girls. They're weird. Any other questions?
So majority of my notes come from being in lecture, obviously, and her lecture videos, but those lecture videos are like 3 years old. And
True.
So from the gram-negative cocci, there's the Neisseria mozzarella and then like the Thio margarita. But not until recently, there was like another Thio margarita that was discovered, which is like the Magnify.
Yeah.
Are we need to know that?
So yeah, always know the most recent information. So February 23rd, 2022 was the discovery of the largest bacterium ever. It was found on mangrove leaves and it was 2 centimeters long, which is very, very big for a single cell bacteria. So yeah, that is know the most recent information. The lecture videos are a little bit old. So I believe that information, yeah, the information about the like 2022 discovery one was in the PowerPoint, in the bacteria PowerPoint. So know that, yeah, when I took the class, they were still trying to come up with a name for it because we went into class and it was like the day after. Like I remember Dr. Kokesh was like, this was announced like 2 days ago or over the weekend, and I just added it to this PowerPoint. But they're still trying to come up with a name for it. Don't know if they've come up with a name for it by now, but the suggested name was Thiomargarita magnifica. So I believe that most recent one, like the 2 centimeter one, is on the PowerPoint. So know that there might be like an extra credit question about that. I think it was an extra credit question last year, but I can't remember exactly. So
Yeah. Okay. So should we just know the Thiomargarita, like the one that's with an N that was like found? And,
yes,
like,
know the original one as well. Like know the Thiomargarita namibiensis. Yes. Know that one too, because it is historically, like scientifically significant. But also know the more recent information too.
Okay. Thank you.
Yeah. Other questions, you guys can type in the chat or you can ask.
Is gamma radiation and ionizing radiation the same thing?
Gamma radiation and ionizing radiation. Let me flip over here because, again, don't want to give you guys wrong information. So ionizing radiation and gamma radiation. No, I think they're different things. Yeah, so radiation. So, oh, wait.
Doesn't gamma
Yeah.
like go in iodizing though?
I was about to say, yeah, no, I'm looking at it now and it has like gamma rays and X-rays. Ionizing radiation is gamma rays and X-rays are used. So yes, this is why we check our notes. They are kind of the same thing. I think gamma radiation, gamma radiation is a type of ionizing radiation, but not all ionizing radiation uses gamma radiation, if that makes sense. It's kind of like the all squares are rectangles, but not all rectangles are squares. So yeah, gamma radiation is a type of ionizing radiation, but not all ionizing radiation uses gamma radiation. X-rays are also used. And can anyone tell me what type of control of microbial growth radiation is? Is it physical or chemical? Or other? Physical, yes. It is a type of physical control of microbial growth. So yeah, they're kind of the same thing, but it's more that gamma radiation is a type of ionizing radiation. Other questions? If you don't have any more questions, I can go over a couple of things that I did specifically for this exam. The first one I can think of while we're talking about control of microbial growth is the list, the list of most resistant to least resistant. I believe it's like at the end of the PowerPoint on microbial growth. It's the one that has most resistant prions, and then going down you have endospores of bacteria, and then mycobacteria, cysts of protozoa, vegetative protozoa, gram-negative bacteria, fungi, including most fungal spores, viruses without envelopes, gram-positive bacteria, and then the least resistant is viruses with lipid envelopes. Come up with a mnemonic to memorize the order of those, either from most resistant to least resistant or least resistant to most resistant, whatever works for you, because there were some questions like, what is more resistant, vegetative protozoa or viruses without envelopes? There were questions similar to those. Or like, it's like it would give an example of each. It would give an example of a specific type of bacteria. Like it would give an example of like a gram-positive bacteria and a gram-negative bacteria and say which is more resistant. So memorize that. And then my tip that I always give for these exams, especially since you can't backtrack, is when you start the exam, but have a blank sheet of paper with you, show that it's blank to the camera in Respondus. And then before even looking at the first question, just write down all of your mnemonics. They're really helpful to help you not second guess yourself in things. So come up with a mnemonic for microbial growth characteristics. And then after you begin the exam, your paper's blank before then. After you begin the exam, do a brain dump of your mnemonics and the order of most resistant to least resistant. It's helpful, just always helps me having a visual. Yeah. So that one. And then the tips for bacteria, it's really just repetition, going over them again and again, knowing like, and it helps to break it down too. It's difficult to learn everything about all of the bacteria all at once. So if you start off like, okay, I'm just going to learn which bacteria are gram-positive and which ones are gram-negative, that's helpful. And so then once you're comfortable with knowing which ones are gram-positive and which ones are gram-negative, then you can say, okay, now with the gram-positive ones, I'm going to learn their shape. And so then you separate them into shape, and then gram-negative ones into shape. It's a really long way of doing it, but it's helpful because I know for me, like when I had all of the information written on one flashcard for one bacteria, I was like, this is too much to memorize about one bacteria all at one time. So separate it out so that you like learn their gram stains on one day, learn their shapes on another day, learn their oxygen requirements on another day. It's just more helpful that way to split it up so you're not overwhelmed. And it also helps to not confuse the information, I guess. Other things, can I explain R factors? So yes, R factors are I was actually reading about those before this review session because I remember those confused me a little bit as well. So let me find real quick. Okay, so R factors. Yeah, so R factor stands for resistance factor. They're basically genes that code for something that helps a bacteria be more resistant to antibiotics or to being destroyed. Like it could be a gene that codes for a slime layer. Like, so if it gets this R factor, this resistance factor, like if a little bacterial cell, and its R factors are transferred generally through conjugation, I believe. So a little bacterial cell comes in contact with another bacterial cell, and this second bacterial cell says, hey, I have this slime layer and I have the ability to tell you how to make a slime layer. Do you want that? And the little second bacteria says, yes, I do. And so the first one that has a slime layer gives the R factor, the resistance factor, to the second little one. So the second one can make a slime layer. And a slime layer makes a bacteria more sticky, so it can stick to surfaces. It makes it harder for like macrophages to grab them. So an R factor is a resistance factor, just anything that makes something more resistant. R factors are the HFR ones, right? So okay, HFR stands for high frequency recombinant. So kind of, they're a little bit different. They, yeah. No, they're slightly different things. Yeah, HFR, high frequency recombinant, while an R factor is resistance transfer factor. So yeah, they're kind of different things. HFRs, it's a cell that has the ability to like transfer, or it has the gene that would give it the ability to transfer information, but it's in its DNA. I would definitely also bring questions about R factors and HFR to Dr. Kokesh's review session as well, because I feel like she can explain it in a much better way than I can. I know what they are. Like, do you ever have those moments where you like know what something is, but it is hard to explain? Like, I think she has a much better, clearer way of explaining the nitty gritty of conjugation, like with HFRs, F plasmids, and R factors. Yeah. All right. Do we need to know the little circle graph on her PowerPoint? Little circle graph. Which one? Like,
It's like near the end, and it's like R factor type of plasmid. It's like origin of replication, and then like the RTF is like near the pilus and the conjugation protein.
I mean, I don't think you'd need to know the graph to label things, but the graph is helpful in a visual of that whole situation
how it
too. So
works.
yeah, knowing how it works, but you don't need to like memorize the graph to like label which is the R factor, which is the point of origin. So yeah, no, that is less important to know. Like, you don't need to know the be able to label the picture, I guess, but know how it works.
Does that also apply for like the transcription and translation part?
The pictures or the
Yeah, the pictures.
Yeah, no, I don't believe, I mean, again, I haven't seen the exam, but based on what I remember from last semester, I don't think there are pictures of like of transduction versus conjugation versus transformation. I think it's more, it will describe, like it'll be like, which type of genetic transfer is like requires a bacteriophage, and then you will say that is transduction. So it's less the pictures. Like, I don't think it'll be as much the pictures in the PowerPoints, more so the definition of how it works. And then it's like, okay, which one is this? Which one requires a sex pilus conjugation? So yeah, I don't think there are as many images in this one, but then again, I have not seen the exam. I'm just trying to remember from last semester, but to be fair, last semester was years ago, or it feels like. But yeah, no, I don't think it's as many pictures. Good question though.
Thank you.
Any other questions? We're closing in on 5. We can also go until this Zoom like closes out. No, I said we'd go to 5. We can go to 5. So any other questions? I'm trying to think of any other study tips I had for this one specifically. Mnemonics are helpful. The thing I said earlier about make sure to focus on fungi, even though it's a shorter unit. Know the main facts about fungi. Know how to know the different names. Know how to tell the different types of hyphae from each other. Know the different types of asexual spore production. That thing that my brain completely short-circuited on during the Kahoot. Know those better than I do after a semester. Those are important to know the difference between. So here, it's, yeah. So asexual spore types. There are 2, there are 2 major types of asexual fungal spores. There are, I'm going to butcher the name, conidiospores, and there are sporangiospores. So know the difference between those 2 types. And then within those 2 types, there are 4 different kinds of conidiospores, and there's only 1 kind of sporangiospore actually. So that makes it a bit easier to remember too. So 5 total that you need to know. 1 is a sporangiospore. The other 4 are conidiospores. know what makes them different. So I feel like a lot of this class is the main concepts, knowing and calling them fun facts makes it a little bit more fun to study too. Know a fun fact about each main topic. Know that like chlamydiocondia, I have my notes open here, is a thick-walled spore formed by rounding and enlargement of a hyphal segment. So knowing like, just know like pick what thing is the main thing, because it's a lot of information and it can be overwhelming to study for that. But if you just pick one thing that is I feel like this is the most important information about this bacteria, or I feel like this is the most important information about this type of spore, or I feel like this is the most important information about this type of microbial growth, focus on one. And then if you have time in studying, try to find a second one, like try to find what the second most important thing is, because it is a lot of information and it does feel like it can be overwhelming sometimes because of the sheer amount of information there is. But if you narrow yourself down to picking what you think is the most important information about something, it makes studying a lot more manageable and cuts down the information a lot. And she will ask about the big points, like how things work, how things are different from each other, and like main points about things. So know which bacteria causes a blue-green pigment and is seen in burn patients. There's a lot of other information about that bacteria, but if you know that it causes a blue-green pigment, that will probably be one of the main things about it. One of the things, like knows things that define things and make them separate from each other. If that makes sense. It's know the main points. And it helps to not get overwhelmed with the amount of information there is. Any other questions? All right. So, yep. It's bacteria, microbial growth, fungus, and microbial genetics. Do you think the next exam will be easier than this? I'm trying to think about what's on the next exam. Viruses are very interesting. So you do viruses on the next one. They're fun, but they come with their whole other range of things. You learn about the immune system, different types of antibodies. And I feel like in terms of information wise, there's more to memorize in this exam and the next exam and the last exam. I feel like the first exam is more how things work. Like you have the chemistry, like chemistry in the first exam. They're each difficult in their own ways, but they are each so you kind of have to approach each of them with a different studying tactic. I would definitely say memorization is the most important studying tactic for this exam, and it's a lot of information to memorize, but that's why narrow it down to the big main points, and come up with mnemonics, even if the mnemonics are silly. Sometimes if they're silly enough, they're really easy to remember. And so they don't need to make sense, just whatever works best for you. And yeah, I'm trying to think about what else is on your next exam. It's viruses and the immune system, and what else? It's fun stuff. Oh, yes. So specific diseases of specific body systems. So like diseases, there will be a unit on diseases of the respiratory tract. There will be a unit on diseases of the integumentary system, one on STIs. So, and this unit with bacteria, will help you in that unit too. Each unit builds on each other. So it gets easier to study in a way for each one, because you're like, oh, I know this information. I had to learn this for the last exam. And then you can build your new information off of the information you already know. So like knowing the different types of oxygen requirements, you needed to know those for the first exam. So in the second exam, now you need to know the oxygen requirements of those bacteria, but you already know what those oxygen requirements are. So helps in that way, like it builds on itself. So don't know if it'll be easier, but it's still interesting information. The prokaryotes and eukaryotes from the last exam. Yeah, and now you do it more in depth for fungi and bacteria. So bacteria, they're your prokaryotes, fungi, they're your eukaryotes. Yes, remember that fungi are remember that fungi are eukaryotes, even though they can be unicellular. Yeasts are still eukaryotes. They still have organelles. They, just because they're one cell, like you can have a single-celled eukaryote. So important thing to remember, not all unicellular organisms are prokaryotes, but your bacteria are. So yeah, but fungi are not. Remember that prions aren't actually any kind of anything. Like remember that prions are just proteins. They don't have any kind of genetic material. They're not bacteria. Prions aren't bacteria. They're not viruses. They're just proteins that get warped and can therefore warp other proteins. They're terrifying, very interesting, and they're the hardest things to kill. They caused mad cow disease. So there's your fun fact about prions that you can know, might be extra credit. I don't know, just speculating. But yeah, so know your fun facts, mnemonics, make flashcards of the bacteria and separate what days you're studying which things, like which days you're studying the gram stains, which days you're studying the shapes, and so on and so forth, just to separate it out so you're not as overwhelmed. But yeah, it is 5. Thank you guys so much for hopping onto this second portion of the review session. I know it's not as fun because there's not a Kahoot, but I will be sending that Kahoot out as well, so you can practice on your own. And feel free to email or text me about any questions you have leading up to the exam. You guys got this. It's going to go swimmingly. But yeah, go to Dr. Kokesh's review session, also super helpful. Would recommend asking her about the specifics of conjugation because I cannot explain it for some reason. But yeah, you guys are going to do great. Have a great rest of your weekend. Study, but also don't stress yourselves out too much about this. Take some time to breathe and drink water. But yeah, thank you guys so much for coming and have a great rest of your day. Bye guys.